Hello, my name is Jude Robinson, and I am delighted to welcome you to today's IPC Partners IPC Journal Club. We have Dr. John Otter, who will be talking to us about Mpox Clade Ib, the next pandemic. And his paper that is picked today is by Marzinio, titled Epidemiological Quantities for Mpox Virus Clade 1 from Historical Data with Implications for Current Outbreaks from the Democratic Republic of the Congo. Before I hand over to John, who will talk to us about that paper, we've just got a usual introduction slide. Those of you that have been with us before will have seen this, but if you haven't, it was just to tell you a bit about what the Journal Club is about. And what we want to do really today is to provide some insights into a topical IPC published paper, which John has picked for us. And we're going to generate lots of discussion. So please do use the chat function. And if you have a question that you'd like me to pose to John after his talk, please do use the Q&A function as well. So this is a great opportunity for continuous professional development. It's going to really exercise your critical thinking skills. It's going to help you to build confidence for independent research and study. And we've already fostered a massive network of enthusiasts, and we are delighted that you are now part of that. And amongst all of that, we're going to have a little bit of fun as well. All of these journal clubs are recorded, and they'll be made available to you after the event. And I will now hand over to John.
Well, thank you, Jude. Thanks for joining us today. It's a pleasure to be here. Jude, just to let you know, if it's a really hard question, then I might be sending it to you. So you might not only be asking the questions, you might be answering some as well. So I hope that's okay.
I don't think we agreed to that.
Well, you know, I said only the hard ones. The easy ones I'll take care of, don't worry. So yeah, thanks for coming, everyone. So Mpox obviously still very topical for any of us working sort of towards the front lines of healthcare facilities around the world. Conscious again, we've got an international audience on the call today. So what I'm going to be talking about will be mainly focused on a United Kingdom context, but I'm hopeful that the principles that we work through will be relevant in whichever part of the world you happen to be working in today. So why I chose this article? Well, Mpox remains topical. We've got an ongoing outbreak of Clade 2 Mpox in mainly the gay, bisexual, and other men who have sex with men populations. We also have a more recent and emerging, really, outbreak of Clade 1 Mpox centered in the DRC in Africa. And that's really the focus of our discussions today. We've had several alerts around those working in the NHS in particular, around how to deal with patients with query Mpox, both Clade 1 and Clade 2. Most recently, only a couple of weeks ago, we heard the news that Clade 1b Mpox have been identified in the UK in a couple of hospitals in London, and that's still an ongoing developing scenario. So I like this article because it's a real epidemiological paper. The methodology gives us nice, detailed information about some of the key parameters of transmission of Mpox, focusing on Clade 1, which will allow us to make some informed judgments and take a view about the transmissibility and even the pandemic potential of the Clade 1 Mpox. I hope this isn't a bit of a plot spoiler, but the title of Journal Club today is Mpox Clade 1b the next pandemic? Well, the short answer is no. I really don't think it's going to be. Although, you know, never say never. So let's dive in. So before I get into the paper for today, just to give you a little background on the clinical features of Mpox, it is a viral infection with an incubation period of around 5 to 21 days. It's usually mild and self-limiting, with most individuals affected recovering within weeks without the need for healthcare. However, severe illness can occur. So characteristically, symptoms are initially a fever and headache, followed 1 to 5 days later by a characteristic rash. And in the top right-hand panels, you can see images of how this rash looks and develops on different skin types. And much like chickenpox, it's contagious until all the scabs have fallen off and there's intact skin underneath. To understand the issues with Mpox, we need to just think a little bit about the clades involved. So Mpox is genomically split into two clades, clade 1 and clade 2. Clade 1 is really, until very recently, restricted almost entirely to Africa, with some historic outbreaks of infection. We've had this recent emergence of the clade 1b clone, which has spread a little bit further. Clade 2 was also historically restricted more or less to a different part of Africa, but a couple of years ago, we began this change in the epidemiology of Mpox Clade 2 with the emergence of Mpox causing infections all around the world. And this really began in 2022, and since then we've had something like 10,000 cases worldwide. And you can see from that age distribution, it's relatively young, almost exclusively men, and it's almost exclusively gay, bisexual, and other men who have sex with men. So it's spreading worldwide, but in a really defined demographic of our population. The epidemiology of clade 1b Mpox is different. Interestingly, earlier this year, the WHO declared a public health emergency of international concern because of the worry that this clade 1 Mpox would spread outside of the DRC into other parts of Africa and potentially around the world. So between January and this month this year, there's been a big upswing in cases, something like 25,000 cases, although it's very difficult to know exactly how many there have been, given gaps in reporting, with something like 700 deaths. So not an insignificant case fatality rate, but lower than has been reported historically for clade 1 Mpox. And that's against a background of around 1,000 cases in 2022 and 3,000 in 2023. So it's a massive upswing in cases. Unlike historic clade 1 Mpox, clade 1b has been in a broader geographic area with an even split between men and women, which is in contrast to the clade 2 outbreak. And there's been some genomic diversity within the outbreak when you look closely, and frequent involvement of sex workers as well. We have seen some clade 1b Mpox cases reported outside of Africa now in Sweden, Thailand, and now in the UK, all of which have had travel links to affected African countries. And UKHSA and other public health agencies, ECDC and CDC, continue to assess the overall risk to the public from Mpox as low. So let's jump into the study for today then. So this is an epidemiological evaluation involving modelling of key transmission parameters of Mpox, and it's all based on historic clade 1 Mpox data, and more about that later as a limitation of the study. So to understand the study, we need to think about the terms involved. So it looked at the incubation period, the generation interval, the serial interval, and the time varying reproduction number. So the incubation period is the time between exposure and the appearance of symptoms. The generation interval is the time between the initiation of infection in an infected person to the initiation of infection in their infectee. And the serial interval is the time between successive cases in a chain of transmission. The time varying reproduction number is a similar idea to the basic reproduction number. So the average number of patients in an infectious disease will spread to from a single case, but it's kind of averaged out over time. So R0 is the basic reproduction number, RT is the time varying reproduction number. Now, I'm conscious there's a lot of information on this slide and a lot of definitions. It's probably easier to look at this graphically. This is a nice graphic, actually not from today's study, but from a different one related to a different virus, but the principles are the same. So you can see there the incubation period is in the blue, so the time from infection to symptoms. The generation interval is from the beginning of infection to the beginning of infection, and the serial interval is from the beginning of symptoms to the beginning of symptoms. If you'd kind of do the maths on those different variables, you can make some inferences on the relative importance of pre-symptomatic transmission. And again, we'll come back to that a little bit later. So the data sets used to make these estimates for incubation period was from 15 patients with sufficient detail on disease onset, and then the serial interval and generation interval were estimated from two data sets, one with 32 household transmission infector-infectee pairs, and one with 11 hospital transmission infector-infectee pairs, and those were from a single hospital outbreak in the DRC. And this is what the results came out as. So the incubation period as a mean was around 10 days. No surprises based on the kind of published ranges of incubation period for Mpox. The serial interval was a mean 17.5 days for household transmission, a little bit shorter for hospital transmission. Again, probably no surprise given the close proximity of patients in a hospital setting. The generation interval was very similar on both counts, 17.2 days for household, 11.3 for hospital. And 3 different epidemiological methods were used to estimate these parameters. I won't go into the details of them, not least because I don't really understand them, but all of them yielded comparable results. Looking at those parameters together and plotting them out as it were, pre-symptomatic transmission was felt to play a minor role, accounting for less than 20% of transmissions. Importantly, the time-varying reproduction number for the 2024 outbreak in the DRC was estimated at less than 1 overall. And that's really significant because if a reproduction number is less than 1, it means that a disease is not going to spread through a population. If it's more than 1, then it will perpetuate through a population. It was found to be the reproduction number was found to be more than 1 in one DRC region. Now, the reasons for this aren't known. The authors of this paper gave a possible reason for this difference to be the higher concentration of sex workers in this mining region. So that's one possible hypothesis as to why the transmission of the same virus would be higher in that particular region of the DRC. Moving on then to limitations and points for discussion from this paper. So as I mentioned, the incubation period is no surprise, perhaps in fact almost exactly in the middle of that estimate range. Mpox is transmitted via a variety of routes. We know that, but it does seem that very close contact and often physical sexual contact is required in order to initiate spread. So it's not a surprise really to see these parameters of transmission on the low side. The epidemiological parameters estimated in this study from historic Clade I data present to us an infectious disease with relatively low transmissibility when we compare it with some other viruses and other infectious diseases, and we'll do that in later slides. I think the major limitation of this study for us to discuss and take away really is that the data sets used as a basis for these estimates are pretty limited. So the incubation period was estimated from a set of including only 15 patients, and the information on serial interval and generation interval in hospitals is based on only 11 infector-infectee pairs from an outbreak in a single hospital. So you really do question the generalizability of these findings. And it's without doubt true that a data set including a wider variety of settings and scenarios and countries would make these estimates a bit more generalizable. One particular challenge when estimating reproduction numbers is limited surveillance and/or limited diagnostic capability, because in a low resource setting, it's natural that only the most severe cases will present in healthcare and will be detected, and the relatively minor cases won't be detected. So that means that R is often underestimated if many secondary cases are missing. So it may be that the reproduction numbers associated with Mpox are rather higher than the literature would have us believe. So how do we put these numbers in context with our understanding of other infectious diseases? Well, if we look at this table, which is a bit of an amalgam of various different publications, we can see the R0 of Clade I Mpox is typically less than 1, and that's consistent with this study that we've been looking at today. Whereas the R0 value associated with Clade II Mpox, Clade IIb Mpox, so the 2022 outbreak, is certainly above 1, something like between 1 and 3. Now, if we can compare that with something like measles or chickenpox, those R values are in double or even triple figures for measles. So the transmissibility of Mpox, even the more transmissible clades, is orders of magnitude lower than some of the more infectious agents that we're used to dealing with. We see a similar pattern if we look at secondary attack rate. So for the clades combined, the secondary attack rate of Mpox in a household setting is something like 0 to 10%. So you could be living with somebody with Mpox, and 9 out of 10 people would not catch Mpox. That's what these data are telling us. Whereas with measles, that's turned on its head. And if you're living with somebody with measles, you are more than likely to be exposed, and slightly less so for chickenpox. This is the secondary attack rate for household transmission of the SARS-CoV-2 virus that causes COVID. It's from a systematic review and meta-analysis, and what you can see is that the secondary attack rate changes over time. So it started off at around 22% for beta, 29% for delta, 36% for alpha, and 42% for omicron. So you can see that as the virus changed over time, it became more infectious, and although this study doesn't tell us, less virulent. And importantly, in this study, the secondary attack rates were lower when either the index case or the contacts were vaccinated. So what does this mean for us then, working in applied infection control? Well, the Mpox Clade Ib outbreak continues in the DRC. Further cases with travel history to the affected area are likely to present in our healthcare systems in the UK and elsewhere. Therefore, we need to have a really carefully defined clinical pathway in place for staff to manage the initial presentation of patients who may have Mpox symptoms, both Clade 1 and Clade 2, especially in the emergency department and in sexual health services. The main take home from the epidemiology in this study is that compared with other infectious diseases, the transmissibility of Mpox is low and very closely linked to intimate physical contact. So the short answer is, as I mentioned, is very unlikely to be the next pandemic. So Jude, I'm going to stop there. I'm going to stop sharing my slides, and let's see what questions we have had come through.
Thank you very much, John. That was really, really interesting. Just while people think about their questions, I'll start us off, if that's okay. So you talked a little bit, John, about the epidemiological parameters of Mpox. Do you think that will change over time?
Well, that's a really good question. I think we've got some evidence from Mpox as a virus that it has changed over time. So we've seen Clade 1 and Clade 1b develop. We've seen Clade 2 and Clade 2b develop over time. And particularly in the case of Clade 2b, we see a bit of a step change in transmissibility. So that same virus has mutated and has become more transmissible. We've looked at the data with household transmission with SARS-CoV-2. In that it has become more transmissible over time, admittedly less virulent, and that's often the case with these viruses as they mutate and change. They become more transmissible, but often less virulent. So I think viral mutation is guaranteed, and they will change over time. There's often a trend towards things becoming a little bit more transmissible over time, but I still don't see Mpox turning into the kind of virus that is going to cause a major pandemic of public health threat because of that requirement for close physical contact in order to spread. Not like being coughed on by somebody with SARS-CoV-2.
Thanks, John. That's really, really helpful. So you talked a bit about some of the limitations of that study. You spoke about the sort of the data sets that you felt just perhaps weren't as robust as they could have been. So what would you have liked those data sets to have looked like?
Well, yeah, that's kind of the golden question, isn't it? So I think let's answer that question by thinking what was the problem with the data sets that we had. So starting with the incubation period, we had 15 patients from an old outbreak of Mpox Clade 1 in the DRC in Africa, for whom there was sufficient information to say that's when we thought you were exposed, and that's when your symptoms had onset. I think the first thing to say is that that's quite a small data set with only 15 individuals involved. You'd like to see more cases in order to get a robust estimate in terms of confidence intervals around that mean. I think you'd want to see more variety in setting. This was in a single country in Africa that doesn't have some of the advanced healthcare systems that we're used to seeing today that may influence some aspects of the transmission parameters of Mpox. And then I suppose similarly for the generation interval and serial interval, the data sets were based on, I think it was for the 30 odd infectee pairs for the serial interval, and in household settings, and something like 11 or 12 in hospital settings. So again, same challenges for those. Really, you want to see more. You want to see a bigger number of patients involved, so you had a more confidence around the estimates. And I think you want to see more settings as well, particularly with that hospital set, because the different hospital settings around the world are so different in terms of their physical infrastructure, proportion of single rooms, hospital cleaning and disinfection, staffing ratios. All of these things are going to have quite a big impact on the transmission potential.
Thanks, John. That's really helpful. So a question we've had, so that generation interval, then we don't hear about that a great deal, do we?
Yeah.
What's your thoughts on that and using that as a sort of a measure?
Yeah, I suppose we don't hear about generation interval or serial interval too much. They're quite technical terms, aren't they? But I suppose that they're both measuring something fundamentally quite similar, which is how quickly the disease is turning over. It's almost like if you, I'm a microbiologist, I'm going to go back to the lab, Jude, and think about a microbiological analogy. It's a bit like the time it takes for a cell to divide in the lab, and how quickly you're going to get from 1 to 1,000,000 with an overnight culture, those kinds of questions. And that's the sort of information that generation interval can give you. And these are the kind of estimates and parameters that are plugged into the big disease transmission models that start to say, well, we are beginning to see transmission in the community. All of the kind of public health surveillance information that came through during COVID. This is the kind of baseline epidemiological stuff that we need to put into those models to give us an accurate picture of transmissibility and transmission potential as well.
Thanks, John. That's really helpful. So you've kind of alluded to this a little bit in some of your sort of comments that you've made. But how do you sort of see this outbreak playing over time with the Clade Ib?
So the Clade Ib outbreak in DRC, looking into my crystal ball to tell the future. I mean, obviously I don't know. Who knows? Nobody can tell the future. But I think it's probably going to carry on a similar trajectory than it's on at the moment. I don't think it will get particularly bigger than it is in terms of disease and outbreak trajectory. Unless there's a significant mutation in the virus, in which case it might. I do think we are going to see more imported cases, though, as the pool of infected patients in Africa grows. So yeah, I definitely think we should all be prepared and ready on the front lines and briefed our teams to say, these are the symptoms, this is what we do when we have a suspected case.
That's really good advice. Thanks, John. We've had a question a bit about sort of distinguishing the difference between Clade 1 and Clade 2 clinically. What can you tell us about that?
Yeah, well, I suppose the answer is you sort of can't really, because from what I understand, there's not really a difference in the way that Clade 1 or Clade 2 Mpox presents. You still get this characteristic initial fever, headache, kind of non-specific initial symptoms, and then something like 5 days later, this characteristic rash emerges. So I think at that stage, it's very difficult. You couldn't really tell whether it's Clade 1 or Clade 2. So it will take time to get that genomic information to tell you with certainty whether it is clade 1 or clade 2. So I think at the moment, the advice is that we use the travel history as the main point to distinguish between clade 1 and clade 2. So if the patient has been traveling to a country of interest in Africa, then we'd assume that they're a clade 1 until proved otherwise. And if they haven't, then we'd assume that they're a clade 2. And we are still continuing to see clade 2 Mpox turning up from time to time in sexual health and in emergency departments. So that they are, I think the number of cases has slowed, but we are still seeing them. So I think frontline teams are getting used to seeing clade 2 Mpox, and I think that's helped really when it comes to the threat from clade 1, and probably got people a bit more comfortable with the disease and seeing how it presents.
Yeah, no, definitely. Another question we've had. So this is a good one actually, John. Nice clinical one for you. So what PPE should I use to care for a patient with query or confirmed Mpox?
Well, that's a good question. So at the moment in the UK, we have a distinction of whether or not we consider Mpox to be a high consequence infectious disease, based on some of the defined criteria that I mentioned earlier. So it's a clinical case definition to say whether or not we manage initially as a clade 1 high consequence infectious disease, Mpox, or whether we manage it as clade 2. If there's been travel history and symptoms at the time of presentation, then you'd manage it as a clade 1 Mpox, and therefore as a high consequence infectious disease. And the answer as to exactly what PPE you wear will depend on exactly where you work, because at the moment there's a bit of a variety of practice of what would be the frontline PPE to care for a patient with a suspected high consequence infectious diseases. So the National Infection Control Manual in England recommends the full kit in that scenario. Whereas it's accepted at this stage if your organisation does something slightly different in terms of maybe a slightly lower level of PPE for the initial care and diagnostic questions and testing of the patient, then that may be the case where you're working. So it would be a case of speaking to your local infection control team and getting advice.
Thanks, John. That's really helpful. So you talked a little bit about on one of your slides around the transmission of the clade 1b. How do you think that sits alongside? You did bring up a couple of different pathogens and infections. What do you think that in terms of that transmission potential? It seems from what you've said is that it's quite low. Do you think that might change, or are there other worries that we need to have, do you think?
Yeah, I think right now we'd have Mpox clade 1 pretty low down in terms of transmissibility compared with other infectious diseases, with that requirement for that close physical contact. Whereas for something like measles, which I would see as like the top of the tree, it really transmits in a fully airborne way, right across the room, as it were. We're really not seeing that epidemiology for Mpox. It could change. It could become more transmissible. Who knows? But even if it does, I think it will still remain relatively low in terms of transmissibility.
That's great. Thank you. I can't see any other questions coming through at the moment. I don't know whether anyone's got any thoughts that they want to put in the chat or the Q&A.
All right. What I will do then is let me reshare these slides and we'll show you what's coming up in the next journal clubs. So, okay. So next journal club is actually next week. It's going to be Phil talking about antimicrobial surfaces, time to rethink surface disinfection. As you can see from the slide there, we've filled out the schedule for journal club until towards the end of January now, with some great events coming up. You can go to IPC Partners website for the schedule for future journal clubs to access recordings from previous journal clubs, and also to subscribe whenever we schedule a new one. Or you can go directly to the YouTube channel to get recordings from previous journal clubs. As Jude mentioned at the start, IPC Journal Club is hosted by IPC partners. And all that remains to say is a very big thank you for joining us today. And we hope that you can join us next time.